Hello and welcome. Today, we're stepping into our laboratory to conduct a practical experiment to test for food molecules. So far, we've learnt about the activity of enzymes in our lessons. We understand that enzymes are specialized proteins that catalyze biochemical reactions, making them essential for various bodily functions. We've covered that enzymes help to break down large insoluble molecules into smaller soluble ones that can be digested by the body's cells. Now, building on this foundation, we introduce our new practical where we will test for different biomolecules, specifically carbohydrates, lipids, and proteins using qualitative reagents. Testing for these biomolecules will help us grasp how enzymes work in breaking them down during digestion, and how we can detect their presence in different foods. Before outlining the practical, let's take a moment to consider its importance. Understanding how to test for carbohydrates, lipids, and proteins in food isn't just an academic exercise. It's used regularly in the world of nutritional science. Nutritional science and dietetics rely heavily on these tests to determine the nutrient content of food items. By engaging in this experiment, you get some hands-on experience with techniques that dietitians and food scientists use daily. This practical knowledge enriches your perspective on nutrition and will help you understand the ways in which we monitor the biochemical content of food, in order to understand its nutritional value. This experiment also provides insight into biological molecules and processes. Carbohydrates, lipids, and proteins are fundamental components of all living organisms. Understanding their structure and function is crucial for comprehending broader biological processes, such as metabolism, energy storage, and cell structure. Using qualitative reagents to test for these molecules gives you a deeper appreciation for how our bodies gain these molecules in order to be used for these functions. Performing these tests will also improve essential laboratory skills. This includes taking precise measurements and preparing solutions and recording qualitative, descriptive measurements of results. These hands-on skills reinforce the importance of accuracy and proper technique in research. Finally, this practical encourages you to hypothesize and draw conclusions based on observable data. You will learn to interpret color changes or other reactions that indicate the presence of specific biomolecules. This fosters critical thinking and trains you in data analysis. These analytical skills are not only valuable in the laboratory, but are also broadly applicable to problem-solving scenarios in everyday life. Now we know the importance of this practical, let's dive in and explore the world of carbohydrates, lipids, and proteins through qualitative testing. Let's start by gathering our materials. Sam, what do we need?
So for this practical, we need a pestle and mortar, a food sample, a beaker, a glass rod, a conical flask, a funnel and filter paper, test tubes, a lab book, a water bath, and numerous solutions to test the food. For this experiment, we will be using Benedict's solution, iodine solution, biuret solution, and ethanol.
For this practical, it's crucial to maintain a precise and clean setup to ensure reliable results. We'll walk through each test methodically, ensuring that our laboratory environment remains organized and tidy. We will begin by breaking up a small piece of the food sample selected using a pestle and mortar. We should now have smaller food pieces that can be used to make a solution. Next, we need to transfer the resulting pieces into a beaker of distilled water. Now, stir the mixture with a glass rod to help dissolve some of the food into a solution that we can use for testing. As the final part of the preparation stage, we need to use a funnel lined with filter paper to filter the solution to remove any solid bits of food. Filtering the mixture in this way removes solid particles that might interfere with the colorimetric changes observed in the tests. Now we have a liquid sample of the food. We will conduct several tests on small volumes of this sample to test for different biomolecules. Let's move on to our first test. In this stage, we will use Benedict's solution to test for reducing sugars in our food sample. Firstly, prepare a test tube with 5 cm cubed of the food sample. Next, let's add the Benedict solution to the food sample in the test tube using a pipette. This will turn the solution blue to begin with. Next, set up a water bath and maintain it at 75 degrees Celsius. Place the test tube in the warm water bath for 5 minutes, which will activate the test and allow us to detect whether reducing sugars are present in the food sample. Now let's look at our results. If reducing sugars are present, the solution will change from blue to green, yellow, orange, or brick red, depending on the concentration of sugars. Adding Benedict's solution and heating up the solution will therefore enable us to detect the presence of reducing sugars. Make sure to make a note of your results and colour change for the food sample in your lab book. Let's move on to our next test. This time, we will be using iodine solution to test for the presence of starch. Start by transferring 5 centimeter cubed of the food sample into a new test tube. Then, add a few drops of iodine solution. Next, gently shake the test tube. If starch is present, the solution will change colour from brown-orange to either blue-black or black. Therefore, we can use iodine solution to check for the presence of starch. Remember to make a note of your results in your lab book. Time for our third food test. In this test, we will use biuret solution to test for the presence of proteins in a sample. Start by putting 2 centimeter cubed of the food sample into a test tube. Then biuret solution and mix gently by shaking the tube. If protein is present, the solution will change from blue to pink or purple. On to our fourth and final test. In this stage, we will test the food sample for the presence of lipids by using the emulsion test using ethanol. Start by placing 5 centimeter cubed of the food sample into a test tube. Next, we need to add a few drops of ethanol to the test, followed by a few drops of distilled water. Finally, shake the test tube gently until the sample has mixed together thoroughly. If lipids are present, a cloudy emulsion will form. Throughout these steps, the changes in colour and formation of emulsions are clear indicators of the presence of carbohydrates, lipids, and proteins. Let's now move on to the analysis of this practical. Firstly, we need to categorise our results based on the observed physical changes and what these indicate. Here is a table summarising the colour change or physical change we can expect to see in light of a positive test. For Benedict's test, we can see a sequence of blue to green or yellow or brick red based on increasing amounts of reducing sugars. The iodine test will show a clear change from brown-orange to blue-black in the presence of starch. The biuret test will show a transition from blue to pink or purple if protein is present. Finally, the emulsion test shows a formation of a cloudy emulsion if lipids are present. With this table recorded, we can identify the results of food samples. For each food sample, conduct the test and record the colour or physical change observed. Once all of this is recorded in a table, we can draw accurate conclusions about the presence of different food molecules. For instance, in the case of a potato, the iodine test produces a change in colour and forms a black solution, thus indicating a strong presence of starch. Furthermore, although the tests are qualitative, some can be partially quantitative. For instance, the extent of colour change in the Benedict's test can suggest the relative concentration of reducing sugars in different samples. You can use a colorimeter or a colour chart to measure the level of colour change and corresponding concentration of reducing sugar quantitatively. In today's lesson, we conducted a practical experiment using qualitative reagents to test for various carbohydrates, lipids, and proteins in food samples. We started by preparing our food sample before conducting a Benedict's test, iodine test, biuret test, and emulsion test to detect the presence of sugars, starch, protein, and lipids respectively. This practical reinforces our understanding of biochemical molecules in food and the methods scientists use to identify and study them. Moreover, it highlighted the importance of careful methodology and the direct application of theoretical principles in experimental contexts. Watch the rest of the GCSE biology course at Sophos Education.